exponential amplification does not take place in PCR with single-stranded nucleic acid molecule and one complementary primer. And I've shown you how this works out here in this example here. For downstream experiments, the RNA is first converted to cDNA for two reasons. Number one, RNA is extremely unstable, and therefore, if you want to perform any experiment on RNA, it has to be first converted to cDNA, which is a lot more stable. And one of the reasons why the genetic material switched from RNA, which was largely believed to be the original genetic material, to DNA was the stability of the DNA. And then, of course, the second important point is that exponential amplification can only happen when you have a double-stranded DNA and two primers to start your PCR reaction. So therefore, when you work with RNA, first the RNA has to be converted to cDNA, and after it is converted to cDNA, it is then taken forward for quantification using exponential amplification. So this is what we're going to see next. There are two types of polymerase chain reactions that we're going to talk of in the next slide. One is what is known as reverse transcriptase PCR, where we are going to convert the mRNA sequence into the complementary DNA sequence, and this complementary DNA sequence can then be amplified. And while amplifying, you want to see real time what is the level of amplification, which is basically done by what is known as real-time PCR. So we're going to see how this works out in the next coming slides. So here is an mRNA molecule, and this could also actually represent the entire transcriptome or all expressed messenger RNAs in a cell. And you know that most of the messenger RNAs in a cell end up in a poly A tail, and that poly A tail can be used as a handle to amplify and convert this mRNA into a double-stranded DNA. The process is known as formation of complementary DNA. And basically what we use here is an oligo-DT primer. And then with the help of reverse transcriptase, you are able to add complementary DNTPs depending on the nucleotides present on the mRNA sequence here. So this is how you synthesize the first DNA strand. So what you now have is an RNA-DNA hybrid. And in the next step, you basically remove the RNA strand. And in place of that RNA strand, you synthesize a DNA strand, which now makes it completely double-stranded DNA. We'll not go into the details of how this is done, except for the fact that usually you use RNase H that can partially dissolve the RNA strand, and the steps that are left allow you to extend and add complementary nucleotides here with the help of DNA polymerase. What you can also do is you can add an adapter at the 3' end of the first strand, and then use a complementary primer to that adapter to synthesize the second strand of DNA. The mRNA strand is replaced by a complementary DNA strand here, right? So this is now finally your cDNA, which is basically a double-stranded DNA with the same sequence as was present in the RNA. Now this DNA, this molecule can be used for amplification. So I'm sure you remember from the previous slides that if you start with X molecules of DNA and perform PCR for N cycles, the total DNA molecules on completion of PCR is X into 10 to the power N. So here in this case, again, depending on what is the original number of mRNA molecules for a specific gene, you would get as many number of complementary DNAs to begin with. So you now use primers to amplify the cDNA. And what you would also be interested in would be to see in real time what is the extent of amplification that happens for your cDNA molecule. So to be able to quantify the extent of amplification as it happens is the essence of real-time PCR. So what we do essentially is we use fluorescence to have a real-time check on what is the extent of amplification that is happening. Most commonly, the dye that we use is sybr green. Sybr green can basically bind to both single-stranded DNA and double-stranded DNA, but preferentially it binds to double-stranded DNA. So sybr green has an absorption maxima or can be excited with blue light, and then once it is excited, it can emit as an emission maxima of 520 nanometers, which is basically the green light. So the extent of fluorescence that comes upon excitation of the sybr green after every cycle can give you an idea of what is the extent of amplification that is happening. And also, if you have two different samples, you can compare the extent of fluorescence coming from each other. So here is your initial molecule of cDNA. Here is your starting of the PCR. You have one cDNA molecule, and the sybr gene is bound to it, but it may not be, but the system may not detect it. Then as you move on, after the completion of every cycle, the DNA doubles up, and therefore the amount of cybergreen that bounds to these DNA molecules also increases and comes into the range of detection. And then, of course, as you move along, you have further increase in the amount of double-stranded DNA. After completion of every cycle, the amount of DNA was going to double up, and therefore the amount of cybergreen that binds also doubles up, and that allows you to detect the increase in the number of DNA every cycle as we move into the exponential phase of PCR. So the intensity of fluorescence increases with every cycle and can be monitored in real time, allows for quantitation and comparative quantitation of DNA. So if you have two different samples with different concentrations of DNA, their comparisons can be done. One can also find out what was the initial difference in terms of the amount of DNA present in the two samples by using the real-time PCR. So what are the essential components of real-time PCR? So most of the components are exactly the same as what we had begun with when we were doing the normal PCR. So you'll have DNA or cDNA, you'll have primers, you have polymerase, you have magnesium ions, you have DNTPs. But the most important thing along with all of this, the difference with the conventional PCR is that you also have a fluorescence reporting system. In the initial days of real-time PCR, we use sybr green. Now, of course, we have more complex reporting systems like TACMAN probe, beacons, and scorpions. TACMAN probe, beacons, and scorpions will be discussed elsewhere. We stay with sybr green for now. There are certain important parameters that one needs to check when you're doing real-time PCR to give an idea of comparison of two samples. One of these values is what is known as the CT value. The CT value is the number of cycles taken to reach a specific amplification level, and that amplification level is actually reported by the fluorescence of the fluorescent marker that you use. And then the other important value is delta CT. Delta CT is the difference in the number of PCR cycles taken to reach the same level of amplification by two different samples. So if there are two samples that are comparing, the ratio of A by B can be given by 2 raised to power delta CT, right? Where delta CT is the difference in the number of cycles taken to reach the same amplification level, and 2 raised to power delta CT will give you the actual ratio of the initial concentrations of A and B. So now let's follow it better with an example. So here is a simple example of quantitative PCR. You have two samples, A and B. The initial number of molecules for A is 4. The initial number of molecules to begin with is 1. For B, the final number of molecules that we're looking at is 64 for both A and B. The number of cycles required by A to reach 64 level is what is known as CTA. Likewise, the number of cycles required for B to reach 64 molecules is what is known as CTB. And how do we monitor the levels of amplification? We monitor the levels of amplification by measuring the fluorescence that comes from on the completion of the PCR cycle. So let's now calculate for A first, and then we calculate for B. And since we're starting the experiment, we know that the original ratio of A to B is 4:1, because A has 4 double-stranded DNA molecules, while B has 1 double-stranded DNA molecule at cycle 0 of the PCR. So this is 4/1 or equivalent to 4, right? So now we calculate the CTA value for sample A. So here is solving for A. So 4 into 2 raised to power CTA equals to 64, which means 2 raised to power CTA equals to 16, or CTA, or the CT value for A equals to 4 here, right? Next, we solve for B. So again, you know, the final number of DNA molecules at cycle n is given by the formula x into 2 raised to power n. So here x equals to 1. So 1 into 2 raised to power CTB equals to 64, which means CTB equals to 6. So now the CTA is 4, CTB is 6. So now we calculate the delta CT. What is delta CT? The delta CT is the difference in number of PCR cycles taken to reach the same amplification level by 2 different samples. So delta CTAB, which means you're comparing A and B together and you're calculating delta CT here, equals to 6 minus 4 equals to 2 here. So now if you put it to the formula of finding the ratios of the initial samples, initial concentration of molecules in the two samples. So here you are, ratio of A to B equals to 2 per delta Ct. We know that the delta Ct value is 2. So therefore, putting delta Ct value here, it gives you the ratio of A by B equals to 4, which is the same as what we know because we started with the same number of molecules here. So this is what is known as quantitative PCR. Quantitative PCR allows you to quantitate the amount of DNA that is getting formed every cycle, and using CT and delta CT, you can go back and calculate the initial ratio of two samples to begin with. So this is your quantitative PCR. And I'm sure you already know that the primary test for checking for SARS-CoV infection is the real-time PCR. So this is how you do your real-time PCR and quantitate the amount of DNA that is present in your test sample with respect to a control sample. So with that, we come to the end of the lecture on PCR. There is a lot more to PCR than this, of course, and as and when we have time, we'll discuss more about PCR, particularly the tinkering that you can do with polymerase chain reaction that allows for a lot more innovative methods of biological assays. So we'll discuss those as we move along during the course.